Explain in detail the mechanism of resveratrol extracted from mulberry fruit in detoxifying aflatoxin carcinogens. Resveratrol is a phenolic compound found in mulberry fruit that has high antioxidant activity. Many studies have shown that resveratrol plays an important role in protecting the body from carcinogens such as aflatoxin B1, AFB1, a toxin produced by Aspergillus flavus and Aspergillus parasiticus fungi. Here are the main mechanisms by which resveratrol from mulberry fruit helps in the detoxification process of aflatoxin. 1. Antioxidant effect. When aflatoxin B1 enters the body, it is converted by the liver enzymes cytochrome P450, 450 cypriot pounds, especially CYP1A2 and CYP3A4, into AFB1-8, 9-epoxide. It is a toxic active substance that can bind to DNA, causing cancer. Resveratrol can inhibit the formation of reactive oxygen species, ROS, caused by this transformation process and reduce damage to DNA, RNA and proteins. Reduces liver damage from lipid peroxidation. 2. Induction of detoxification enzymes. Resveratrol stimulates phase 2 detoxification enzymes that play a role in detoxification, including glutathione S-transferases, GSTs. Helps bind AFB1 epoxide to glutathione, GSH, to reduce toxicity and increase excretion. UDP glucuronylyl transferases, UGTs, and sulfotransferases, salts. Helps bind glucuronic acid or sulfate groups to toxic substances to make them more water-soluble and excreted in bile or urine. 3. Inhibition of cytochrome P450 enzymes. Inhibition of phase 1 enzymes. Resveratrol can inhibit the activity of CYP1A2 and CYP3A4, reducing the conversion of AFB1 to AFB1 epoxide, which is a toxic and mutagenic form. 4. Protect DNA and inhibit mutations, anti-mutagenic and anti-carcinogenic effects. Resveratrol can inhibit the binding of AFB1 epoxide to DNA, DNA adduct formation, reducing the risk of mutations. Stimulates the expression of genes involved in DNA repair and apoptosis in damaged cells. 5. Anti-inflammatory effect. Reduces the production of inflammatory cytokines such as TNF-alpha, IL-6 and NF-kappa B, which are factors that stimulate cancer. Summary. Resveratrol from mulberry helps detoxify aflatoxin by reduces the production of toxins from phase 1 enzymes, stimulates the elimination of toxins by phase 2 enzymes, protects DNA from damage, reduces oxidation and inflammation. Explain the mechanism of occurrence of liver cancer, stomach cancer, kidney cancer, gallbladder cancer, colon cancer, breast cancer, ovarian cancer, colon cancer, etc. From the carcinogenic substance aflatoxin contaminated in food. Contamination of aflatoxins in food, especially aflatoxin B1, AFB1, which is the most toxic and carcinogenic type, plays an important role in inducing cancer in many organs of the body through complex mechanisms at the cellular, molecular and genetic levels, which can be explained as follows. 1. The main mechanism of carcinogenicity from aflatoxin 1.1 bioactivation AFB1 is metabolized in the liver by the enzyme 450 cypriot pounds, especially CYP1A2 and CYP3A4, into toxic intermediates, namely AFB1-8, 9-epoxide. AFB1 epoxide is highly sensitive to binding to biological molecules such as DNA and proteins. 1.2 DNA adduct formation. AFB1 epoxide binds to guanine bases in DNA, forming AFB1N7 guanine adducts. Mutations in important genes, such as the p53 tumor suppressor gene, especially at codon 249, AGG right pointing arrow AGT, are associated with liver cancer. 1.3 Oxidative stress AFB1 induces the generation of reactive oxygen species, ROS, damaging cell structures and DNA. 1.4 DNA repair suppression disrupting DNA repair mechanisms, causing accumulated damage. 1.5 Epigenetic modifications such as methylation of tumor suppressor genes, mRNA changes. 2. Organ-specific carcinogenesis by aflatoxin 2.1 liver cancer, hepatocellular carcinoma, HCC, it is the most aflatoxin-related cancer. AFB1 epoxide binds to DNA in liver cells. Right-pointing arrow mutates the p53 gene. Right-pointing arrow abnormally regulates the cell cycle. Right-pointing arrow cells grow relentlessly. If combined with hepatitis B virus, HBV, they synergistically cause cancer. 2.2 Gastric cancer AFB1 produces ROS in the mucosal cells. Right-pointing arrow chronic inflammation. Right-pointing arrow DNA damage. Aflatoxin-induced changes in the microbiota affect immunity and inflammation. 2.3 Colorectal cancer undigested AFB1 in the intestine is absorbed and exposed to the intestinal mucosa, causing damage to intestinal mucosal cells and disrupting the microbiota. Affects the APC, CRAS and p53 genes. 2.4 Renal cancer aflatoxin and some of its metabolites are excreted in the urine, thus reaching the cells in the kidneys. Accumulation of toxins. Right-pointing arrow chronic kidney cell damage. Right-pointing arrow mutation. 2.5 Gallbladder cancer exposure to aflatoxin excreted in bile. Right-pointing arrow oxidative stress in gallbladder mucosa. 2.6 Breast cancer. Although not the primary target of aflatoxin, studies have shown that AFB1 affects estrogen and stimulates estrogen receptor positive cells. It may also inhibit the function of BRCA1, BRCA2. 2.7 Ovarian cancer. The mechanism is similar to breast cancer, DNA damage and hormone receptor stimulation. AFB the 1st of May alter the expression of genes related to ovarian cell division. 3. Synergistic factors. HBV or HCV infection increases the chance of developing liver cancer from AFB1. Weakened immune system increases the risk. Poor nutrition, e.g. lack of antioxidants, lack of protein, reduces detoxification ability. Long-term exposure to aflatoxin, even in low doses, if